Hello, everyone, and welcome to Talk Nerdy. Today is Monday, June 26th, 2023, and I'm the host of the show, Cara Santa Maria. And as always, right here at the top of the show, I do want to thank those of you who make Talk Nerdy possible. You've probably noticed that there are fewer ads than recently. We're still doing some of those programmatic ads, but fewer what we call host-read ads. So these advertisements that I partner with different companies to kind of develop and work on together. That has always been a big heartbeat of the payment structure of the show. This is how I pay my editor, how I pay my incredible assistant who I couldn't live without, Noelle Dilworth, and also keep this profitable for myself as well. There are just fewer ads across podcasts. You're probably noticing that left and right. And so because of that, Patreon has become much more important, and we're all kind of leaning on that a little bit more. I really do want to thank those of you who are already patrons of the show, and encourage you, if you're in a position to do so, that you can pledge as little as like $1 an episode. I think as little as $2 an episode gets you an ad-free version of the show. This week, I want to thank the top patrons, and they include Daniel Lang, David J.E. Smith, Mary Neva, Brian Holden, David Compton, Gabrielle F. Jaramillo, Joe Wilkinson, Pasquale Gelati, and Ulrika Hagman. Thank you so much. If you are interested, all you've got to do is visit patreon.com/talknerdy. All right, so I had the opportunity to have a fascinating chat today with Dr. Barbara Rae-Venter. She's got quite the bio, and I really want to make sure that I don't do her any disservices. So let me tell you about this brilliant woman and some of the really important things that she's done. So she has both a JD and a PhD. We'll talk about that. She's the president and a co-founder of Firebird Forensics Group, which is a 501c3. So it's a nonprofit corporation that assists law enforcement with identifying suspects in violent crimes and identifying unidentified human remains. She's the director of Investigative Genetic Genealogy at Gene by Gene, which is the parent company of Family Tree DNA. And she's going to talk about that a bit on the show. But basically, she is an investigative genetic genealogist. She's going to tell us what that means, but it's just really, really interesting. So she's a retired intellectual property attorney. So that's where the JD comes in. But in undergrad, she studied psych and biochemistry, and then she got a PhD in biology later on. And kind of the reason that many people might recognize Dr. Rae-Venter's name is because she identified Lisa Jensen, who was abducted in infancy and abandoned by her abductor as a 5-year-old. So by identifying her mother, the New Hampshire State Police were able to solve the murder of the Allenstown Four in New Hampshire. Subsequently, Dr. Rae-Venter was able to determine the true identity of her abductor and the suspected murderer of those four. She also assisted the FBI and the Sacramento DA's office in identifying Joseph James D'Angelo as the notorious Golden State Killer. Across the board, she's helped out in solving like 70 cases. And because of that, she was recognized by Nature as one of 10 people who mattered in science in 2018, and by Time magazine as one of their 100 most influential people of 2019. What we're going to talk about today is that investigative genetic genealogy and the big role that it played, actually the lead role that it played in identifying the Golden State Killer and getting justice for a lot of the families of the victims. So she's written a new book. It's called I Know Who You Are: How an Amateur DNA Sleuth Unmasked the Golden State Killer and Changed Crime Fighting Forever. So science meets true crime in this one. So without any further ado, here she is, Dr. Barbara Rae-Venter. Well, Barbara, thank you so much for joining me today.
Well, thank you for inviting me. This is kind of an exciting opportunity, so thank you.
Oh, I'm so glad. So we're going to chat for a little bit today about your new book, I Know Who You Are: How an Amateur DNA Sleuth Unmasked the Golden State Killer and Changed Crime Fighting Forever. But of course, before we dive into the story itself, I want to learn a little bit more about you. So you're an investigative genetic genealogist and biochemist. You're also a retired patent attorney, and you've worked with the FBI on a lot of different cases. Let's unpack that for a minute. What is an investigative genetic genealogist?
Well, it's a new occupation. What we do is we use a combination of DNA and traditional family history type research to identify suspects in violent crimes and unidentified human remains.
Okay, this is so interesting because we recently had a guest on who is a forensic anthropologist. And so she was looking at significantly older, maybe not, human remains from a biological anthropology perspective. I can imagine that there's a fair amount of crossover, but you're very much focused on like cellular material.
So the source of DNA for what we're doing, it can be basically any biological fluid. And then, of course, if you're working with unidentified human remains, it can also be bones and teeth. We also can obtain DNA from rootless hair, which is a new technique that's been developed for using with IgG.
Oh, rootless hair. Yeah, because I think any of us who have watched a lot of kind of crime scene television shows were under the impression that you needed that little skin tag at the end of the hair, that little piece of the root.
Correct, and it's turned out not to be true.
Oh, wow, that's fascinating. And then of course the biochemistry component of your training just feeds into that, right? Into the genetic focus that you have as well. What did you study in school?
Well, that's a good question. I actually started out, when I started college, I was actually a French literature major. And I went to a junior college here in Northern California, the College of San Mateo, and then transferred from there to the University of California at San Diego. And so in the summer between leaving the College of San Mateo and going to UC San Diego, I decided to change my major from French literature to pre-med. So obviously that entailed doing a lot of different things than I had been doing in terms of coursework. But that was sort of a kind of watershed moment, I guess. So my actual degree, my bachelor's degree, is a double major in psychology, because actually what I wanted to be was a psychiatrist, and biochemistry. And then from there, I actually had work study money while I was an undergraduate. And so I was actually working in a research lab. It was a biochemistry lab. And I just really got pulled in by doing research. It was just really exciting. It actually was, there's some similarities between what I'm doing now and working on research. It's the same kind of roller coaster emotional ride. Where when your experiments are going well, it's a real high, and of course when they don't work, it's you're really feeling kind of dispirited. So there's some real parallels there. So anyway, so I ended up with instead of going to medical school, or I didn't even apply, I just applied to graduate school, and so I ended up with a PhD in biology. It's actually in biochemistry because there was no PhD in biochemistry at the time. I have the background in biology there. And then my first teaching position, I was actually teaching endocrinology and biochemistry. So I also have a fairly decent knowledge of endocrinology as well.
Oh, interesting. And I detect a bit of an accent. It sounds like you did a lot of your training in California. You also, I'm seeing here in your bio that you also went to UT at Austin in Texas and earned a law degree after all of this scientific training.
Yeah, that's correct. So I guess I like doing different things, and so after doing research for, I guess it was about 7-8 years, I found that I was really interested in bioethics. So actually, when I went to law school, what I was planning on doing was actually bioethics. And there were all kinds of interesting topics at the time. Obviously, this is a few years ago. So the hot topics at the time were things like frozen embryos. How do you appropriately apportion out tissue for pediatric patients. So, for example, if two kids need a liver transplant and you only have one liver available, how do you decide which child gets it? Because it's basically a life or death situation for the child that does not get the transplant. So those were sort of some of the questions. I was just fascinated by the whole concept. So, but then when I was graduating from law school and starting to look for jobs, people would look at my resume and of course they'd see all the science and they'd say, well, you should be a patent attorney. And so I actually didn't know anything about patent law. And so University of Texas actually had some intellectual property classes. And so I took some courses and I actually found it very interesting. And so that was what I ended up doing, was being a patent attorney, and I specialized in biotech type inventions. A lot of the inventions were patenting of genetically modified organisms of one kind or another, anything from bacteria to yeast to plants. So lots of interesting stuff.
Yeah, and it's interesting because there's this underlying thread that does tie it all together, but from the outside, ostensibly it feels like these are kind of like wildly different career paths. I'm curious about then the timing of it all. Were you a patent attorney first and then you got into the investigative work kind of with the FBI, or did these tracks sort of independently follow one another?
So I actually retired from patent law in 2005.
Okay.
And so I was working on my own family history research, and I was using DNA because my ancestors had done some interesting journeys from one country to another. So on my paternal grandmother's side, my ancestors had all come from the north of Scotland via Nova Scotia to New Zealand. And trying to track back from New Zealand to Nova Scotia to Scotland using paper records was very difficult. There were not a lot of records. And so I just decided, okay, I'll just use DNA. So I got into using DNA on my own family history. And any relative that I could corner and get DNA from, I did. So I have about 20 relatives that I managed to convince to do DNA testing.
Oh, wow.
And of course, when I was then working on my research, the matches that I would get, quite often I would get people who were adopted and were trying to find their biological family, and I had no idea how to help them. And so I thought, well, no, this is ridiculous. I should be able to help these people. So I went online and I found a course that was offered by a group called DNA Adoption. And that's actually how I learned to do genetic genealogy, was by taking, at the time it was a 6-week course. And, by the time I got through, I was fairly good at identifying biological relatives for people who were adopted. And it was from that I ended up then getting into IgG, because one of the other things that I did at DNA Adoption is when they learned that I had a science background, they asked if I would help with teaching their class and with answering some of the questions that came in on their webmail. And so there was an email that came in from a Deputy Peter Headley in the San Bernardino Sheriff's Department Crimes Against Children detail. And he had a case that he was working on. It was a woman who is now in her 30s, but she had been abducted as a 6-month-old.
Oh,
And of course,
wow.
she had no idea, anything about her family history, where she was from, who her parents were, anything like that. It's kind of interesting because they had thought she was maybe older when she had been abducted. They didn't know for sure. She was recovered when she was about 5. And so they were actually getting ready to try and do some things like hypnosis and see if they could, recover any memory, because of course there wouldn't have been, because she would have been way too young. But they didn't know that. And then the deputy had seen some information. Actually, I don't think it was the deputy. It was actually the woman's name where she was using the name Lisa. And she apparently had seen, I think it was, do you know, what is it called? Oh, now forgetting the name of the show. That's ridiculous. We'll have to redo.
Describe it. Maybe I'll know.
So anyway, it's who do you think you are, or something like that.
Yeah. The kind of genealogy, like where did your
Right.
family come from? When did
Yeah.
they migrate? Yeah.
So she apparently was watching that, and so she asked the deputy, can we do something like that to figure out who I am? And so he had then sent in an email to DNA Adoption asking if this technique that we're using to help adoptees, could we use that then to figure out who Lisa is? And so I said, yes, it should work. And so I then volunteered to work on that. So that was kind of the beginning of everything.
Oh, wow. So it was like a really personal way in, and you just happened to have done the training and to have the tools available to
Right.
be able to sort of, I guess in some ways would you consider it a career for you, or was it a hobby after you were retired? Like, how do you identify that personally?
Well, so that was in 2013. So I've now been doing this no, 2015, excuse me. So I've now been doing this for 8 years. So I think
Mhm.
it's gone past being a hobby.
Right, right.
I spend, enormous numbers of hours a week doing this. So it's sort of become another career. My son overheard me talking to somebody, and they had asked me if I was, and I had told him that I was retired, and he kind of looked at me and goes, "Mom, I think you're over that."
Yeah, you're back at it. And so just to be clear again, I started to ask and then I got distracted by all the interesting things. But so you were talking about your family history. You yourself are from New Zealand. Is that where you were born?
Yes,
But then you came here. How old were you when you came to the US?
I was a blushing bride of 20.
20. And then you did most of your training here in the US, your
I did
academic training.
Yeah,
Okay. All right. And so now we are, I'm sitting here, I'm looking at your book. Is this your first book?
it is.
That's so very exciting. And so, okay, I know who you are. How an amateur DNA sleuth unmasked the Golden State Killer and changed crime fighting forever. Who, pray tell, is this amateur DNA sleuth?
Who is the sleuth? That's me.
Mm-hmm. And so this happened, was it towards the beginning of the work that you were doing? Did this, like, you consider yourself an amateur because you kind of started on your own, right?
Yeah, it's kind of interesting. One of the reviews, and I'm not remembering which one it was, made some snide comment about that and said it was good marketing on the part of Penguin Random House that I was hardly an amateur. Um, I'm not sure that's quite true. I mean, now I would say, yeah, I'm no longer an amateur. But at the time I started working on the Golden State Killer, yeah, I worked on the Lisa Jensen case, and I'd worked on identifying the guy. Actually, it was the guy who had abducted her, figuring out who he really was. And there was then an odd connection. So there was the guy who had abducted Lisa, he was actually arrested in Contra Costa County. And Contra Costa County is where Paul Holes was, who was working on the Golden State Killer. And so there was some communication between the two detectives. And Detective Headley was, of course, bragging about the fact that we'd solved who Lisa was. And so Paul heard that, and he was all, well, how did you do that? And so next thing I know, I'm getting a phone call from Paul Holes wanting to know if I'll help him on one of his cold cases. And of course, that cold case turned out to be the Golden State Killer.
Mm-hmm. And so maybe you can take a few minutes to inform the audience, those who maybe haven't read up on the Golden State Killer. I think for many people, it might strike a chord like, oh, I've heard that name. Some people might be like, yes, I know all about this case. But for others, they may not know anything about it. So maybe you can kind of fill us in a little bit on just the lore, the history, kind of the general knowledge about the case.
Sure. So anybody who lived in Northern California, more so than probably other places in the country, would definitely know who the Golden State Killer was. I mean, he had a reign of terror from around 1976 for about 10, 12 years. He was responsible for, I think it was over 150 break-ins. So he started out like a lot of these guys do, with break-ins. He apparently particularly liked ladies' underwear. So he would go through people's lingerie drawers and dump stuff on the floor. He earned the nickname the Visalia Ransacker because of the mess that he would make. From there he escalated to rape, and he was responsible for at least 50 rapes. From there, he then escalated to murder. And so he was then subsequently responsible for 13 murders in addition to all the rapes. So, and then he stopped, or at least he apparently stopped. And of course, there's a big question about did he really stop or did he just change his MO? As far as anybody knows, he stopped. So the case then went cold for 40 years.
What was the time span of his when he was active?
It was 1976 through, I think it was 1986.
Okay.
And so, yeah, and I mean, he was just incredibly prolific through that time period. So he as I said, he then just sort of disappeared off the radar. When we caught up with him 40 years later, he's actually living in Citrus Heights in Sacramento, which is where all of the early rapes took place. So he was actually, hiding in plain sight, you could say. So it was a emotionally wrenching case to work on. Just reading through stuff. I also went to the trial and the hearing, and then also the victim statements. And it was just emotionally exhausting listening to this stuff. The depravity in his behavior was just unbelievable. He was a true sadist, and he initially started with just women, single women, and then there was a meeting, I believe it was in Citrus Heights, where one of the guys made a taunting comment about how, he just went after single women. He wouldn't do this if there was a guy home, kind of thing. And the very next case was then a couple. And so what he then did is he would have the woman bind her husband. He often used things like shoelaces. And then what he would do is he would place plates on the husband's back and tell him if he heard any rattling of the plates, he would kill the wife and he would kill the guy. The rapes were repeated and brutal. And then he would, in several cases, he would call people after the fact and he would talk through clenched teeth and say, I'm going to kill you. And there was one woman who she actually ended up getting divorced, moving, remarrying. He tracked her down something like 15 years later and started phoning her and threatening to kill her. So yeah, he was just a terrible person.
Yeah, I mean, I'm obviously very curious about some of the things that you learned about him, especially as he was identified. But even before that, I'm curious, when you first started working on this case, you kind of already referenced a bit how you got involved. But was this a case you knew anything about in advance? Was it something that had already captured your interest and attention, or was it all very new to you?
It was very new to me. I actually was living in Buffalo at the time. So obviously it was huge news in California, particularly in the Sacramento area. And actually he also expanded down to the Bay Area, down to Walnut Creek area. So he was coming down to the San Francisco Bay Area. So people in Northern California were very, very aware of this case. But for people outside the area, yeah, I mean, I don't know that most people really knew about him. For me, it was, yeah, I'd not heard from him. And I think when Paul told me, eventually which case it was, he expected a reaction. And of course, I'd never heard of the case before. So I started, Googling it and finding all of this horrendous stuff because obviously there was a lot of ink on it. And yeah, it was shocking is, I guess, the best thing I can say.
Absolutely. And so I'm curious, and obviously you're not a detective. You're not, you work for the police, but I'm curious why a case goes cold? Is it really just because they've exhausted the leads, they've done all the witness interviews? It was before a time when we had these scientific tools available to us, and they just literally were like, we don't know what to do now.
Correct. So what you had starting in actually would still have been some time period after GSK was active. So I think CODIS became available. So CODIS is the combined DNA index system, which is maintained by the FBI, and it contains DNA profiles for people who have been convicted of various crimes. And here in California, if you're arrested for a felony, your DNA is taken and put into CODIS. So there's, I forget how many profiles are now in the database, but that didn't become available until I believe it was around 1990 or a little later. Prior to that, so when the Golden State Killer was active, the tools that were available were things like matching blood types and looking at various enzymes in the blood. There was nothing that was even close to being as sophisticated as what we're doing.
Right,
So, as
right.
long as he was careful and didn't leave things that we did have ability to detect, like fingerprints, there wasn't very much that they could do. And there were some sketches of him, but if you look at the sketches, he routinely wore a mask, and so the sketches don't even really resemble each other particularly. And so, there really was nothing to do unless you were actually to catch him in the act, basically, or he was to slip up and actually leave a fingerprint someplace, which he didn't do.
Gosh, it's so scary thinking about, just historically, like prior to having like solid forensics. And even there was obviously a fair amount of, I don't think that we knew it was pseudoscience at the time, but a fair amount of pseudoscience in forensics before we had, good kind of reliable scientific investigations. But thinking just about how many terrible things have happened historically that nobody could do anything about because they just didn't know and they had no way of knowing.
Yeah, and I think that's what's really exciting about this tool is that we can now solve cases that were unsolvable. And so there are so many cases. I mean, once the news about the Golden State Killer broke, I started getting emails from all kinds of folks just across the country. A lot of detectives were waiting basically to retire until they'd solved their one last case that was just really sticking in their craw. And so a lot of the guys that I worked with initially have retired. They did their one case, and that was the one that they wanted to solve, and then they could retire. And so a lot of the early cases are really nasty cases. And I think that's something that I really hadn't anticipated was the reaction to working on these kinds of cases. I mean, granted, I'm obviously not being exposed to the same level of horror as the people who were in the field, the detectives, the crime scene people, and so on. But just reading about some of the stuff and seeing pictures about some of the crime scenes, there is an emotional toll, and I really hadn't realized it until I had worked on a case out of Coconino County in Arizona, and the husband of this detective, his sister lives in the same town that I do. And so he and his wife were out visiting. And so we all went out to dinner, which was really kind of fun because the whole family is law enforcement. And so I could actually talk about the cases and, and not be worried about totally offending somebody with the information that you share on these kind of things. And so I don't know what it was that the husband picked up on. So he's a retired deputy police chief, and he teaches classes nationwide to law enforcement on the emotional toll of working in law enforcement. And so he was giving a course here in Monterey, and he asked if I would like to attend the course. He said, he would, just invite me if I would like to do that. So I said, sure. And so I went along. It was a day-long course. The particular class he was teaching, the attendees were all California game wardens. I actually had no idea that a game warden was a sworn peace officer. We apparently have over 400 of them in California, and there were 200 of them at this session. And it's always, of course, a little intimidating when you're at some of these functions because you're sitting there with what are predominantly male, and they're all wearing sidearms. And it's giving presentations to them is also a very interesting experience because they tend to be kind of stoic, and so if you're used to giving presentations to genealogists or scientists or other groups of people who are very engaged in what's going on, and you get a lot of feedback, you get a lot of questions, that doesn't happen when you're giving presentations to law enforcement, at least in my experience. They'll ask questions afterwards, but they tend not to ask questions in the session itself. And you've got this, sometimes hundreds of these mainly men sitting there totally stoic, no expression on their faces, so you can't tell if you're being heard or not heard. I asked one of the detectives about that, and his comment was, they're probably just really excited and thinking about the cases that they can now solve from what you're telling them. And it's a lot to take in. So that was kind of an interesting perspective. One of the things that tends to happen, and I know this happened when I went to law school, and I think it probably also happens to people when they go away to college, you tend to change your friends, and you maybe don't see some of the people that you used to see. I became very aware of that that some of my friends were maybe not quite comfortable around me, knowing that what I was doing. But I think more interestingly was what he pointed out was that you become less tolerant of trivial stuff. And I had actually recently had an experience like that, but I hadn't really realized what was causing the problem. Where I'd been in a social situation where somebody was complaining about something that seemed to me to be completely ridiculous. And normally I would have been, given that I was brought up in New Zealand, you learn to be very polite. You try not to be rude to people. And I would never, ever have left a situation when somebody was, in the middle of a conversation. And I couldn't stand this conversation. It was just, to me, it was just such trivial nonsense. And so I excused myself and went home. And I'm thinking mentally, that is so much BS. And so in the presentation, he goes, and you're going to be in situations where you're listening to somebody and you're mentally thinking that's just total BS. And that was exactly what I had done. And so it was a recognition that, it's been getting to me at some level where, I'm just not willing to, there's so much bad stuff going on out there, and I would rather be spending my time trying to do some of the work that I'm doing than being in a situation where I'm being exposed to that kind of triviality. And so I've become very much more, I guess, selective about who I spend time with. It's become important to spend time with people that I care about and to not spend time with people that I don't find that they're worth spending time with, is I guess the way to put it.
It
That
is
probably sounds a little arrogant, but it really isn't. It's just my priorities have definitely shifted from working on this kind of stuff.
so interesting how this kind of work that can have such an impact and such deep meaning for us in our lives can actually, obviously we affect the work. You deeply affected the work. You did the work and you made the work happen, but that the work can affect us so deeply as well.
Mm-hmm.
I'm actually curious. Maybe we can take a little bit of time to get into some of the brass tacks about, what your contribution was, what you found, how you even went about this. Obviously, this is an entire book. So to simplify it is going to be a bit difficult, but maybe you can share some of the highlights with us.
Sure. Well, I really didn't do anything new. The technique that is, and we all use the same basic technique. Yes, there have been some new bells and whistles that we can use, but the basic technique itself is exactly the same as the one that I learned from DNA adoption back. I took their course, I think it was 2012. And so the techniques have become more sophisticated. There have been lots and lots of fabulous tools that have been developed. But yeah, and we basically all use the same technique. So what we do is you take your crime scene DNA, and as already talked about, it can be a biological fluid, it can be teeth or hair or bones. So you've got a number of different sources for the DNA. So then what happens is a lab extracts the DNA from whatever the biological source is. And then what is created is what's called a SNP file. And a SNP stands for, it's capitals SNP, and it stands for single nucleotide polymorphism. What that is one change in one nucleotide in the DNA sequence. We look at around 700 to 800,000 SNPs. So what a SNP is it's basically it's a mutation. And so since you inherit your DNA from your parents, you get 50% from each parent. And of course, they've inherited their DNA from their parents and so on. So people who were closely related have inherited the same collection of these SNPs from their ancestors. So the more SNPs that you have in common, the more closely related you are. And so if you do a test at Ancestry or 23andMe, they do what's called a SNP array. So they have chips that they incubate your DNA extract with, and then the DNA then hybridizes to the chip. So they can look at the chip, and it's computer read, and there are tags that are put on the chip so that they can then look at, and it can be computer read to see which SNPs you have as compared to other people. And then what Ancestry or 23andMe or Family Tree DNA or MyHeritage, what they do is they then calculate how much shared DNA you have with other people in their database. And then based on that, you can estimate how those people are related to you. So the unit that is used is called a centimorgan. You can think of it as being a linear measure. It's not really, it's actually a probability calculation, but you can think of it as being linear. And so if you have a parent and a child, the amount of matching DNA is around 3,500 centimorgans. If you have full siblings, they have around 2,700 centimorgans. If you've got half siblings, they're around 14, I think it's 1,500, 1,400 centimorgans, and so on. A first cousin would be around 800. So there are tables that have been developed. It's a biological system, so there is a lot of variation. So there can be a lot of quite a broad range in terms of how much DNA still equals somebody being related to, say, a first cousin. But you can use these tables to estimate how somebody is related to you. So what we do is we take then the crime scene DNA, extract the DNA, and then a SNP array is created. So the samples that are used for somebody who's just doing their family history research, they've done a cheek swab or they've spit into a tube. So you've got a very clean, pretty DNA sample. Forensic samples, of course, are not so clean and pretty. They could have come from a semen sample, they could be from blood, they could be from teeth or hair or whatever. And they could also have been exposed to the elements. They could be old. So, for example, the DNA sample we used for the Golden State Killer was from a 30-year-old rape kit. So the DNA in that particular case, we were very lucky. It had been sitting in a freezer all that time, and so it was actually in very good shape. But some of the DNA is damaged, and so you're not dealing with the same kinds of nice, clean samples that somebody who's just doing their family history research would be. So there's going to be some wobble in the numbers when you're looking at these things. So for a forensic sample. So what we do is we then take our forensic sample. If it's damaged or you don't have very much DNA, you might use a technique that's called whole genome sequencing. So whole genome sequencing is actually the original Human Genome Project. They actually use what was called shotgun sequencing. So what they did is they did whole genome sequencing. They sheared the DNA into small fragments and then basically put everything back together again. And that's sort of what ends up happening with the hair, for example. The reason that nobody knew that there was DNA, nuclear DNA in the hair shaft is because it's in little tiny fragments. As the hair shaft grows, the cells actually go through apoptosis, which is programmed cell death, and in that process, the nuclear DNA is chopped up into little tiny pieces. The standard technique for detecting nuclear DNA is PCR, polymerase chain reaction technology. And the spacers that are used in that technique require a minimum size for the fragments of around 150 base pairs. The DNA that's in the hair shaft is only around 35 to 50 base pairs. So way too small for the spacers that are used. And so that was why it was never detected. There was known that there was mitochondrial DNA in the hair shaft, but not that there was nuclear DNA there. So anyway, back to our testing. So you can do, so the whole genome sequencing then is kind of like using the old technique for shotgun that was used for the Human Genome Project. And so you're basically taking the DNA, and if it's not like it's coming from the hair shaft, if it's coming from another source, you then shearing it and then putting it all back together again in order to sequence it. And then from there, you use an algorithm to then pull out all of the SNPs that you would be getting if you had done the chip technology. So now you're just going and looking for those, the address for those SNPs in the genome. And so you're going in and you're pulling those out and then creating a file that would be the equivalent to the file that if you had used a chip. So then you upload these SNP files to either GEDmatch, which is a third-party website. They don't do any testing, where people can upload their DNA profile from wherever they've tested. So if you tested at 23andMe and you've got a cousin who tested at Ancestry, you both upload. You can't obviously see how you match each other because the companies only look at matches within their own databases. So, but if you both upload to GEDmatch, you can then look at your DNA and see how closely you're related. If you're supposed to be first cousins, are you really first cousins, or is there somebody in your family tree that wasn't who they thought they were and you're really not related? So that's what GEDmatch is. And then there's one other company that allows upload of forensic files, and that's Family Tree DNA. And so both of those sites, you then take your SNP profile that you have, you upload it. They use then an algorithm to look for other people within each of those databases who share DNA with your crime scene DNA. And you get a little printout which lists the names and email addresses and then the amount of matching DNA for those people who share DNA with your crime scene DNA. So then what we do is we then take that information and we hope that we've got big matches. So by a big match, I mean somebody who's a second cousin or better. So if you remember, I said a parent-child match is 3,500 centimorgans. A second cousin is around 240. And so second cousins share a set of great-grandparents. And so this is important because once you've built out your speculative family tree for your unknown person, you next have to turn around and do what's called a reverse tree. And so what you know is, since the person that you're trying to identify and some of these people that are in the database share DNA, and let's say they're sharing it as second cousins, then you know that they're sharing a set of great-grandparents. So let's say you've got two people who are second cousin matches to your unknown person. You build out the family tree, you figure out who the common ancestor is. And so what do you know about the remaining person, the unknown person? You know they're also a descendant of that shared common ancestor. So now what you do is you build what we call the reverse tree. You look for all the descendants of that common ancestor. And you know that the person you're trying to identify has to be amongst those descendants, because that person shares DNA with these other two people who we know who the shared ancestor is. They're both descendants of that couple. This person must be also a descendant of that couple. So once you've done that, you then build down. This is actually the probably the more difficult part of doing this whole procedure, because most of the databases, of course, you don't have information about living people. And so
Yeah.
if you're law enforcement, then yeah, you've got access to all kinds of stuff. But if you're people like me, I don't have access to those databases. So we have to use things like obituaries, looking at who are the surviving family members in an obituary so we can add them to our tree. Using things like, there are a number of there are sites that have become available so that you can check people out. So there's places like Spokeo, Being Verified, White Pages. There's a couple of others, Truthfinder. So they are fairly expensive sites, though, and so I have subscriptions to all of those sites. And what they do is when you go in, you put in the name of somebody who is one of your descendants of your couple, and then you can find out more information about that person. So now that you've finally got all the descendants of whoever your couple is, now what you're going to do is you're going to compare those people with what you know about your unknown person. So when you start out, you normally put together a little profile, if you like, of who this person is. So let's see, for GSK, what did we know? Well, the first rape that we were aware of was from 1976. So he was presumably born in, say, 1956 or earlier. What else did we know about him? Well, they were mainly in Northern California. He probably lived in Northern California. In his case, I had also taken his DNA, and there used to be a tool that was available on the GEDmatch site. It's not there any longer, but there used to be a tool there where you could estimate eye color. And in his case, it estimated that his eyes were blue. So we've got those three little pieces of information. So then what you do, and of course you know that the person is male. So I've got my list of descendants. I go through them, and in his case, doing a first pass, looking at just men of the right age who lived in Northern California. I had a suspect list of 9 people.
Oh, wow.
It was a wow. And yeah, I mean, I'm looking at that, and there had never been a suspect list for this guy. And so that was really exciting. So at that point, we decided that it would probably be useful if we could do a consensual sample on somebody to make sure that we really were in the right ballpark. This is the first time that we're trying to use this technique for doing this, and I'm sure the FBI was nervous, and Paul Holes was nervous as to was this for real, that we really had we're closing in on this guy. And so I think I can't remember if it was Paul Holes or Steve Kramer who was the FBI guy. They then contacted a woman that we believed would be a close relative. She's obviously not going to be our suspect because she's female. And so she agreed to test. And so, of course, we're all completely glued to our computer screens while we wait for the results to come back. I must have logged in a gazillion times to see if the results were there yet. And so finally, we got the results back, and she was a second cousin. But what was more exciting for me from a scientific standpoint was she was also an X chromosome match. Now, men only have one X chromosome, and they inherited that from their mother. So we now knew that this person who just tested, that she was related on GSK's mother's side. And it also allowed me to eliminate 3 of the men on my suspect list because they could not possibly be X chromosome matches. So now we were down to six men. And so what the FBI did at that point is we, of course, had one other piece of information. We had the eye color. So the FBI then pulled the California driver's license records for the 6 men that we had left. Only one had blue eyes.
Wow.
And
oh, DNA.
So yeah, I mean, I'm looking at that, and it was so funny because I then got a phone call the next morning. Of course, I sent an email to Steve and to Paul, and said that I thought it was Joseph D'Angelo. And so I get this phone call from Steve the next morning. He goes, "How sure are you?" I said, "Well, as long as we got all the descendants, I'm sure." And I didn't really understand the question because they had the same information that I did. What I didn't know is that there had been a DNA profile done several years before where somebody had actually done a phenotype analysis. And in that analysis, they had come back, and I had not seen this because they wouldn't share stuff with me. And so I had not seen this information. But on this analysis, apparently the eye color had been called out as green and had ruled out blue. And so it's kind of like, but I didn't know that. So that's why Steve is calling me, I guess, is they're looking at different information. And so, of course, things then go silent because I then don't know anything until the day after they've arrested him. I mean, I'm not law enforcement, so I was not privy to then what was going on. So they'd all, of course, raced out, and they did two surreptitious DNA samples. And this is the really important part about IgG that a lot of people don't really understand. So I've told them who I think it is, but nobody's going to take my word for it. They're not going to do an arrest warrant. They're not going to do a search warrant based on me telling them that's who it is. They're going to go out and do a DNA sample. Typically, it's surreptitious. Sometimes they'll end up getting a search warrant to do a DNA sample if for some reason they can't get the surreptitious DNA. They then take that sample, and that then gets compared against whatever's in CODIS. And so they're doing a direct comparison between a sample they've just collected from the suspect and comparing it directly with the crime scene DNA. And at that point, of course, they're getting the results that it's a gazillion to one that it can be anybody else. And so then, in this case, they decided to do 2 samples, even though the first one was a gazillion to one. They wanted another one, which was a gazillion to one, just to make sure they really had the right guy. And then, of course, once he's arrested, they then do another DNA sample to make sure that the guy that they've arrested is the same guy that they've done the comparison on.
What was the crime scene DNA again? Was it hair or was it semen? Do you know what they're comparing it to?
It was a semen sample.
It's from the rape kits. Okay.
Mm-hmm.
And so there's like, now it's like, the probability just gets smaller and smaller and smaller and smaller and smaller that this is a chance alone encounter. And it's just more and more likely at this point. I mean, undeniably likely that this is the guy. What, like, at that point, did you walk away? Were you like, I'm so curious about who this person is? Like, how did you personally, I don't know, like manage your psychological experience when you had done the work and the work led to kind of a definitive match?
Well, obviously, it was very exciting. But I expected that we'd be able to do that, and
Mm-hmm.
I think that's what surprises people. I often get asked that question: is how sure were you that the technique would work? Well, I mean, I'd done it on many people who were adoptees, and it always worked for that. And there's literally thousands at this point of people who are adoptees who have used the technique to identify their biological relatives. So, I mean, I was confident that the process would work. I don't know what level of confidence Paul Holes had or Steve Kramer had, but I was 100% sure that the technique would work. It would be just a case of how long it would take to get decent matches and be able to build out trees that connected so that we would be able to identify a common ancestor. That's sort of the unknown part of all of this. If you've got DNA from a crime scene and it's in reasonable shape, the only question then comes in is to how decent the matches are. I mean, I have a couple of cases right now where the matches are tiny. They're like 20 centimorgans. I mean, that's somebody that's, I don't know, sixth cousin or something. And so those kinds of cases, you then have to try to manufacture your own bigger matches by trying to build out what you can, and then you end up doing a lot of consensual type samples in that scenario, so that you can, basically you're doing a circling technique. You identify the people that you can who are related to that person, and then just start testing their relatives on either side of the tree to then try to figure out where to focus, and then you just sort of identify people that you think should be closer and closer relatives until you then can finally close in on who the person is. We just solved a case about a couple of months ago where most of the matches were around the 40 to 50 centimorgan match. So again, tiny, tiny matches. And there was one that showed up that was, I think it was 80. 80 isn't fantastic either, but in the grand scheme of things there, it allowed us to solve the case.
That's, I mean, obviously you mentioned that like you were fairly confident that this was going to work regardless, but then when it actually works, I can imagine how validating it is, but there's also this like psychological component. Historically you're helping these adopted individuals learn about their ancestry. Now and of course you had been working on some cases as well, but now we're talking about a very vile person who did unspeakable things and justice finally being served. And although there are probably some parallels for you psychologically between the work that you had done and this work, I can imagine that, I don't know, like I'm just curious about what came up for you when you saw the person's name, when you were able to like start seeing the headlines rolling out.
Well, from reading all of the horrible stuff about his cases, I wanted this guy. And so, yeah, it was a feeling like, well, that's where the name of the book comes from. I'm sitting there, I'm looking at it, and it's, I know who you are. I've got you. And actually, I already had the gotcha moment when during the process of doing this, there was another website that we used. It's no longer, well, it was, this is obviously the first forensic case, so there were no rules in place as to where you could upload. And so I had actually uploaded the file to another genealogy site called MyHeritage. They currently will not allow upload of forensic files. So they changed their terms of service after the Golden State Killer case. And so I had actually, it was a company that I had an account with. And so we didn't have very good matches at either Family Tree DNA or at GEDmatch. And so I said to Steve, I've got an account at this company. Do you want me to try uploading the file there, see if we get anything better there? And so I did that, and we got a second cousin match. That was actually when I first said, gotcha. because with that second cousin match, it actually we had built out a tree for one of the smaller matches from Family Tree DNA, and the same people were in that tree as were in the tree for these second cousin matches on MyHeritage. And so that was when I knew we had him, because now we
Yeah,
had him
you
as
already knew.
sister, and now
Yeah.
all we had to do was find all the descendants. I mean, see,
Did you
I say all we had to do. I mean, it's still a lot of work to build down from there, but yeah, I mean, I knew that we could solve the case right there.
did you expect him to be alive?
I'm sorry?
Did you expect him to still be alive?
We didn't know. That was one of the, I mean, there was a chance that he wasn't. And the fact that there hadn't been any new cases linked to him was of concern that maybe he wasn't. For the sake of the victims, we certainly hoped that he was. So no, so it was exciting that that we had somebody that we knew was alive.
And so I'm seeing here that June 29th, 2020 is when he pled guilty, and part of his plea deal basically kept him in life imprisonment without the possibility of parole. So there was no death penalty brought against him at the time. And so he's just going to live out what the remainder of his life. I mean, he's 77 already, but the remainder of his life in prison.
Yeah, so originally, California does still have the death penalty. I mean, there's a moratorium on it, but so that part of the plea deal was that the prosecutors would take the death penalty off the table, and that was, I think, that was just a very good decision when the prosecutors accepted that, because he was old and a lot of the victims were elderly. And so it was important for them. And I mean, the other big thing about the plea deal was that he also had to agree to admit to all of the crimes where the statute of limitations had run. And for many of the rapes, the statute of limitations in California had run. The law has now been changed to change the statute of limitations, but at the time it was fairly short. And so the fact that he then now had to admit, that was huge for the many, many surviving victims of the rape.
Absolutely.
Yeah.
Absolutely. Gosh, I mean, what an incredible amount of work that you put in, but what an incredible reward at the end of that long work, to be able to see justice, justice being served, closure for some of the surviving families and for some of the surviving victims, even. And gosh, I don't want to say it's rare in science, but it's not always in science do we get to do work that's applied and where we actually see the real world ramifications come to fruition. A lot of science is kept in the lab, and a lot of science is, many steps removed from the actual, tangible benefit. And so to be able to see something so direct must have been incredibly rewarding.
It was. I think in part because the crimes were so heinous, and he was. I think also and I think other people felt this also, is his whole demeanor during the hearings, the victim impact statements. I mean, he just sat there pretending to be out of it, which he wasn't. They had pictures of him in his cell doing burpees, and yet he comes in a wheelchair and pretends that he's this frail old man. To me, that was just so insulting to his victims, that he just continued the masquerade.
Yeah. 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 I mean, what a complicated and ambivalent experience for everyone involved. So
Mm-hmm.
listen, I've already kept you at an hour. I don't want to keep you too long. I do always close my my podcast with the same two questions for my guests. But before we get to those final questions, I'm just curious if there's anything that we haven't gotten to, any questions that I didn't ask, or things that you feel like we would be remiss if we didn't mention.
Well, we would be most remiss if we didn't mention that if people want to help law enforcement, that
Mm-hmm.
they can help. If they've done DNA testing at any of the direct-to-consumer companies, that they can download their DNA profile and upload it to GEDmatch and to Family Tree DNA and opt into law enforcement matching. That will help in solving cases. If they haven't already tested, then if they will go test and encourage their friends and family to do the same thing, that would be great.
Excellent. Thank you so much for that advice and for that information. And so, yeah, Barbara, when I have guests on the show, they come from various backgrounds with various scientific credentials, and they write about so many different topics. Like my show really is wide sweeping when it comes to science, and I'm always really curious about my guests' answers to these two kind of fundamental questions. And they're a little bit big picture, they're a little bit deep. So bear with me, and if you need a minute or two to think on it, no worries. But I'm very curious from you personally, when you think about the future in whatever context feels relevant to you right now in this moment. So it could obviously have to do with your work, with the case. It could have to do with your community. It could have to do with something, I don't know, national, global, cosmic, whatever context feels relevant. The first question is, where are you finding your hope and your optimism? What are you, no, I switched the order. We're going to start with the bad one. The first question is, what is keeping you up the most at night? How are you, where are you finding a bit of pessimism, maybe even cynicism? What legitimately worries you? And then on the flip side, when you think about the future, where are you finding your optimism? What are you kind of genuinely looking forward to?
So, one of the things, and this is related to my work, one of the things that I'm looking forward to is that the use of artificial intelligence for assistance in building family trees, that is an extremely time-consuming activity. And there's some folks who are working on tools to be able to do that. And we're already starting to see some of that. I know there have been some tools already developed that the FBI is using. There is also some tools. There's a group called Family Search, which is out of the Mormon Church, and they have been using artificial intelligence to actually build trees from records in the Family Search database. And so they've actually put, I think there are 3 of them at the moment that they've put on their site for people to access. To me, that would be huge because it takes so much time to build these trees, and you could, if you have fast tree building ability, you could then really very easily apply the technique to current cases. IgG was used in the Kohlberger case out of Idaho. People were saying it, but it took 6 weeks for them to identify somebody, and oh, woe is us that it took so long. Well, the reason it took so long is because they were having to build trees. And so if you could build the trees faster, if you've got a particularly hot case that you really need to solve really quickly, if you could use things like AI for helping with building trees, that would be really, really helpful. On the flip side, what is negative? Well, one of the things that we've really run into is people's concerns about privacy. And this, of course, can negatively impact the application of IgG to solving cases. If there are significant restrictions that are put on the use of IgG, I think that would be unfortunate. And I think part of the reaction, a lot of the same, well, let me say it this way, a lot of the same arguments that are being made about IgG and privacy were made about fingerprints. People don't like new technology. They don't like the changed. And so I'm very concerned that it's a misapplication of the concern about privacy because, quite frankly, I think most of us, particularly if you've got Northern or Western European ancestry, your DNA is out there. And so if I was to get a hold of a DNA sample from you, I could probably identify you. I don't need or at least not. I don't need your DNA. I just need some skin cells or something, or some hair, and I could identify who you are. So being concerned about privacy, I think that horse already left the barn a long time ago. So I guess I'm pessimistic that misplaced concerns about privacy may derail the use of IgG, and I think that would be very unfortunate.
Right, that people's individual concerns about their own, I guess, like liberty and privacy would prevent basically certain crimes from being solved, and would keep individuals who are doing harm out in the community.
Correct, and so it is. It's a balancing thing, and I would hate to see the balancing falling too much in favor of individual privacy over the public good, because we also have a right not to be raped and murdered.
Absolutely. And sadly, if that does happen, we have a right for the person who perpetrated that to be put to justice. Yeah. Yeah. Well, gosh, so many insights, so much to think about. Definitely a different approach on this show than we usually take. So I'm just really, I'm happy that we were able to have this conversation, because it's, I don't know, when your book came across my desk, I was like, ooh, this is fascinating. Like, I'm very excited to speak with you. And so hopefully the listeners felt the same way. And I just want to thank you for your time and for all of the hard work that you did, and for putting pen to paper, which is another hurdle, so that you could tell this story in a way that we can learn from it.
Well, thank you. And thank you for inviting me.
Of course, everyone, the book is I Know Who You Are, How an Amateur DNA Sleuth Unmasked the Golden State Killer and Changed Crime Fighting Forever, by Dr. Barbara Rae-Venter. And everybody listening, thank you for coming back week after week. I'm really looking forward to the next time we all get together to talk nerdy.